Hello, greetings. So today we're going to be doing DNA duplication. And so this is going to be how DNA replicates itself. So the first thing that we have to do is we have to unwind the DNA. We have to take this wound DNA and unwind it so the cells can access the DNA inside so you can make another copy. And so the enzyme, the protein that does the unwinding is called helicase. So it'll come along and it'll unwind this protein. It'll unwind the DNA. And then we have the secondary protein called topoisomerase that comes along. And what topoisomerase is going to do is it's going to help unwind the rest of the DNA so that it doesn't snap or it doesn't break. Because helicase is going to be pulling everything apart, it'll make a lot of tension. And so topoisomerase is just there to help relieve that tension. And so once the helicase and topoisomerase are doing their thing, you've got the sugar phosphate backbone right here, and the nitrogenous bases protruding out. And so these nitrogenous bases are connected by hydrogen bonds. And so if we don't do anything, then naturally these bonds will want to come connect back together. And the way that we prevent that is we have these things called SSBPs. And what they do is they just come along, they bind to the nitrogenous bases, and they just prevent that reconnection. So they just prevent those hydrogen bonds from reforming. And so we got these SSBPs on there. And then after that, we have primase that comes along. And so primase is going to come along. And primase is going to go here, and it's going to make a primer, what we call a primer. And so it's going to come along. It's going to take off the SSBPs. And it's going to make a little primer right over here. And the reason that we make this primer is for the next enzyme called DNA polymerase II. And so DNA polymerase II is going to come on in. It's going to find that primer. It's going to bind to the primer. And then it's going to start actually making our DNA strand. So DNA polymerase II is the enzyme that is actually going to make our complementary strand of DNA. And so we're going to take off the SSBPs, and then it's going to make this complementary strand of DNA. And so for this top strand, this is all well and good. This method works. The problem is that DNA polymerase only runs from 3' to 5'. And so this works great for the top strand. We call this the leading strand. And so this strategy is going to work great for the leading strand because you're just going to keep on going. Topoisomerase and helicase is going to keep moving on, keep on winding the DNA, and it can just go on its merry way. The problem is when we get to this bottom strand. And so the difference with this bottom strand is because DNA is anti-parallel, 3' is going to be over on this side and 5' is going to be over on this side. So when primase comes along and puts down a little primer, and then DNA polymerase binds to that, it's going to go 3', which is on this side, down to 5', and make its little strand of DNA. But eventually, it's going to hit a roadblock, because all of this DNA over on this side, this is all bound DNA. This one helicase hasn't done anything to it, you can't access it. So once you get to this side, DNA polymerase can't really do anything else, so it's just going to leave. But we only have this little section of DNA. So when topoisomerase and helicase continue onwards, what primase is going to do is it's going to come along, and it's going to put down another primer. It's going to put down a secondary primer, and then DNA polymerase is going to come along, boop, 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 and it's going to put down another complementary strand of DNA until it reaches the secondary primer, then it's going to leave. And so you see how the bottom one, it's lagging behind the leading one, because the leading one could just go one straight line to make the DNA, but the lagging strand, you've got these segments here. And so what these segments, they're called Okazaki fragments. And so eventually it's going to, make a primer at this replication fork right here, and it's just going to keep making fragments and fragments and fragments on this lagging strand down here. And eventually we're going to get through the rest of the DNA, and we're going to have two strands. Eventually these primers are going to be replaced. They're going to fall off and they're going to be replaced with the DNA. And we're going to have the template strands that we had at the beginning, and we're going to have our new complementary strands, which are going to be these pink ones that we have just made. And then what happens is we have DNA ligase that comes in, and DNA ligase is just going to bind these two together, and so eventually we will have two copies. We'll have this top copy and we'll have this bottom copy, this leading strand copy and the lagging strand copy. And that is how you make, that is how DNA duplicates itself.